I welcome you all to another session. I welcome you all to school or to class rather in this school knowledge without the price. And I'm glad that you follow suit or you keep on attending our lectures as we upload them on this platform. And I encourage you to keep up attending or keep on attending the lectures and make sure you are jotting down notes as the notes are going to help you in the field of health, be it any department you find yourself. So in our previous class, we are talking about carbohydrates. We have talked about carbohydrates, we have talked about lipids, and in this class, we're talking about proteins. And I want to see if you have not watched or if you have not attended those classes, please make sure you go and attend them before attending this, because I'll just be doing a relationship relating the previous lessons to this one. So without much ado, let's go into the topic of today. And today we're talking about proteins. Now, proteins is a simple topic, or let's say it's simple but complex, because we have been hearing of it from elementary knowledge. Even if you don't have a science background, but you should have an idea when it comes to proteins, and at least you should know that when I have a wound, proteins aid in the growth and repairs of tissue. That's the first thing you should think of about proteins. And let's look at it in details. Now, the first thing you have to take note of about proteins is that the first thing, as a scientist, you have to understand that proteins are made up of carbon, hydrogen, oxygen, and additional elements, nitrogen and sulfur. And the next thing you have to take note of is that they form the building blocks of the body. Now, and protein aids in the growth and development. So any abnormality in a protein structure may lead to metabolic alteration or metabolic diseases. Proteins, now in summary, proteins are made up by polymerization of amino acids. The formation of proteins are produced by the polymerization or the fusion of amino acids. Now, when two amino acids combine, it's called a dipeptide. When three amino acids combine, it's called a tripeptide. When it's four amino acids, it's called a tetrapeptide. And when the combination of amino acid is 50, it's called a polypeptide. Now, when a single polypeptide chain contains more than 50 amino acids, it's called a protein. So in summary, a protein is a polypeptide chain which has more than 50 amino acids present in it. That is the summary of proteins. So take note, as source of proteins, you have meat, you have fish, you have eggs, you have spinach, and the rest, which act as edible sources of proteins. Now, without much ado, let's dive straight into the structure of proteins. Let's look at the different structures of proteins, because I made mention of the fact that metabolic diseases arises from the changes that occurs in the protein structure. So let's look at the different structure. Now, the first structure of protein we're talking about is the primary structure. Now, the primary structure denotes the number and sequence of amino acids in a protein. And now the primary structure is a linear sequence of amino acids in a polypeptide chain. So that is what you have to take note of about the primary structure of amino acids and primary structure of protein, and in this primary structure, proteins may either appear as branched due to the formation of disulfide bridges or appear in a circular shape. That's what you have to take note of. Now, the first thing you have to take note of is that first, it denotes the number and sequence of amino acids in a protein. Now, the next thing is that the primary structure of protein is a linear sequence of amino acids, and this linear sequence, proteins may either appear as branched due to the formation of disulfide bridges or in a circular shape. Now, the next thing you have to know about the next structure of protein we're talking about is the secondary structure. And the secondary structure of proteins refers to the folding of the polypeptide backbone stabilized by hydrogen bonds. So the secondary structure of proteins refers to the folding of polypeptide backbone stabilized by hydrogen bonds, and it leads to the production of alpha helix and beta pleated sheets. And I will talk about alpha helices. So I'm talking about the bending of a single polypeptide chain to a right-handed spiral structure. Now, the reason why it is bending to the right is because majority of proteins are dextro-rotatory, not levo-rotatory. Now, if you don't know what dextro-rotatory is, I explained it explicitly in carbohydrates. Then let me just give a summary. Now, we'll talk about when I was dealing with carbohydrates, I talked about the optical activity of molecules. And when we talk about the optical activity of molecules, we are looking at the ability of molecules to rotate plane polarized light either to the left or to the right. So any molecule that rotates plane polarized light to the right is a dextro-rotatory molecule, and if it's to the left, is a levo-rotatory molecule. Now, the reason why we say proteins are Majority of the proteins are dextrorotatory. The reason why they form an alpha helix is because they have the ability to rotate plane polarized light to the right, making it dextrorotatory as opposed to level rotatory procedure. That is that about alpha helix. Now we'll talk about beta pleated sheets. These are sheet-like arrangement of polypeptide chains, either in a parallel structure or an anti-parallel structure. Now the third structure of proteins we're talking about is the tertiary structure. Now we'll talk about the tertiary structure. We're talking about the fusion of the alpha helix and the beta pleated sheets by weak interaction. So that's what I have to take note of about the tertiary structure. It's so simple and so easy to understand. Now, the last structure of protein we're talking about is the quaternary structure, which consists of more than one polypeptide chain. Now, depending on the number of polypeptide chain, proteins may be either a monomer, meaning it contains one polypeptide chain, a dimer, meaning it contains two polypeptide chain, a tetramer, meaning it contains four polypeptide chain. Now, we'll talk about a homodimer. A homodimer contains two of the same polypeptide chain. A heterodimer contains two different types of polypeptide chain. Now, I'll talk about polypeptide chain. We're talking about amino acid. We're talking about amino acid. You may have tryptophan, you may have lysine, and the rest. So when it's made up of just a single amino acid in that polypeptide chain, it's called a homodimer. When it's made up of different amino acids, it's called a heterodimer. That's what you have to take note of. The next thing now we're looking at is the classification of proteins. Now, proteins are classified into different categories based on their functions, their solubility, and all the like. So we'll be exploring those different categories. Now, the first category we're looking at is classification of proteins based on their functions. Now, based on their functions, we have some proteins which are called catalytic proteins. And examples of catalytic proteins, we have enzymes. Now, well, if we go back, I've done a lesson on enzymes, so I expect all my students to go back to that lesson and attend the class. I've explained how proteins aid or how proteins functions as enzymes. So catalytic proteins, we have enzymes. Transport proteins, we have hemoglobin, we have myoglobin, we have albumin. Now, hemoglobin has been explained explicitly in the course of hematology, medical hematology. This course is medical biochemistry, so I urge all my students to go and get the lesson. Now, regulatory proteins include insulin. Regulatory proteins or hormones include insulin. Protective proteins include immunoglobulins. Now, we'll talk about protective proteins. We'll talk about the proteins which aid in the build-up and the maintenance of our immune system. Now, that is that about classification of proteins under functions, or classification of proteins based on their functions. Now, let's look at classification of proteins based on their composition and their solubility. Now, there are two types of proteins. We have simple proteins and conjugated proteins based on their composition and solubility. Now, other simple proteins, we have albumin. And one can say of albumin is that, first of all, it maintains osmotic pressure. Now, talking about osmotic pressure, it prevents the autolysing of cell by eradicating free radicals. So, and also aids in the transport of substances throughout the body. It's soluble in water. The first thing, it maintains osmotic pressure and it aids in the transport of substances throughout the body. Now, the next type of simple protein we're talking about is the globulins. Now, globulins are insoluble in water as opposed to albumin, which is soluble in water and they form soluble salt solution. And under the globulins, there are three different types. We have the alpha globulin, which aids in the transport of substances like hormones, enzymes, and vitamins. We have the beta globulins, which aid in the transition of iron and cholesterol. And we have the gamma globulin, which forms the immunoglobulins that ensures or that aids in the build-up and maintenance of the immune system. And under conjugated proteins, which is another type of proteins under the category of the solubility of proteins. Conjugated proteins, the first type of conjugated proteins we have is the glycoproteins. Now, we'll talk about the glycoproteins. We are looking at when proteins combine with carbohydrates. Now, and when the carbohydrate component is more than 10%, it's called a mucoprotein or a glycoprotein or a protoglycan, sorry, when it is less than 10%, it is called a glycoprotein. Now, and this glycoprotein, they aid in the transfer of substances into and out of the membrane, because there is a question that I asked when I was talking about the cell membrane in the introduction of this lecture. I asked the question: the cell membrane is lipid. The cell membrane is lipid-like in nature. So how come molecules which are not lipid in nature have access to the cell membrane? You notice that glycoproteins play a vital role. Now, if you want to know more about glycoproteins, I'll urge you to go back and attend that class. And I want to encourage all of my students to refer this page to others. You can't just be enjoying this knowledge alone. Make sure you refer this page to others, to your colleagues, to your classmates, so that they can benefit from the same knowledge that you are benefiting from. This knowledge may just be what someone needs to score an A grade in that course, in that department, and this knowledge may just be what someone needs to get to that first class level. Now, so that is that about glycoprotein. The next type of protein we're talking about are lipoproteins. Now, we're talking about lipoproteins. We're looking at lipid molecules which are being attached to the protein molecules. Now, we're talking about lipoproteins. Most often, we're relating it to cholesterol. Most of them relate to cholesterol, which they aid most people, and the function that they do is the transfer of molecules, transfer of lipids, either to the heart, either to the liver, or from the liver, depending on the nature. And they also play a key role in the transfer of substances throughout the cell membrane. Now, the next classification of proteins is the classification of proteins based on their shape. Now, based on their shape, we have the globular proteins, which they are easily soluble. An example of globular proteins, we have albumin, we have the globulins, which are explained earlier. Another type of proteins based on the shape, we have the fibrous proteins, which are elongated and needle-like, and their solubility is minimal, and they aid in flexibility and absorb friction at joints. Example, you have collagen, you have elastin, and you have keratin, which is the main protein in the nail or the fingernail. And now the last class of protein is the classification of protein based on their nutritional value. And under their nutritional value, we have two types of proteins. We have the complete proteins and the incomplete proteins. Now, we'll talk about the complete protein. We are looking at proteins which have all the essential amino acids, or all the essential amino acids, that's the first essential amino acid, in their correct proportions, as opposed to incomplete proteins, which are termed incomplete because they lack one or more essential amino acid. That is just what you have to take note of in summary. So that is that about the lecture on proteins, and I will encourage you to like, to share, to subscribe, and to refer this page to someone. As our tradition demands, there is a package for everyone who has followed suit to the end, and make sure you send in your responses as soon as possible to that email. I can't wait to receive your responses, and if you have a question, make sure you drop it in the comment section, or you can send it through the institutional email, and we are going to respond to you as soon as possible. Don't forget to like, don't forget to share, don't forget to subscribe. Thank you.